Your YouTube, how it started, it says one of the links is how to stream on YouTube Live.
What's up, Sub 3? That's Jill. Burp, burp. What's up, Matt? Nice to see you again. We're going to start this stream in a little bit. How's your days going? Mine's chilling. Sub 3 says, hi, Jill. For the 8 people that are in here, did you guys finish your CRISPR experience? Beep, beep, beep. Sub 3 fighting my technology? Gay? What? What's up, Chris? Yeah, 4 o'clock. 4 o'clock. Can you guys hear me? Yeah? Testing. Matt, you finished the P Dusk P Dawn one. How was it? Easy, hard, meh? Chris hasn't started the last month yet. It's okay. Plenty of time. Julie's still behind. Hope to finish before Biohacker 2 starts. Yeah, so Biohacker 102 starts May 2nd. Oh my gosh, what day is it today? It's May 17th. So in like 2 weeks. The CRISPR experiment realistically takes so you have to grow it overnight, and then you have to transform it the next day, which takes about actually it takes about a little bit, because you have to transform it and then you have to grow it up for 4 hours. It could take you 2-3 full days. Yeah. Sub 3 says long science weekend for me. Hell yeah. Weekends are all about science. Dave Brister, you can hear but with echo. Maybe it might be the room. Echo, echo, echo. Sorry. Echo? Echo, echo? Shouldn't be the case. Do we have a... No. Not on. Second mic is on. Let me see. Hello? 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 We're streaming through something different because YouTube decided to fuck us and not do Google Hangouts anymore. So we're streaming through a separate streamer, which then links to YouTube. So I'm sorry if there's an echo. Yeah, sorry. I'm not a technical person. Ugh. Okay, I will just start chatting with you guys. So, sadly, this is our last class. This is our last online Biohacker 101 class for this class. I'm really sad to see you go. It's been 4 months of awesomeness, just hanging out with you and just chatting with you online, doing experiments with you, and doing lots of genetic engineering. That's Pete. He takes care of all of you. Beep, beep. Pete, turn around. Come here. Say hello. Yeah. Yeah, so Pete takes care of you. Yeah, I'm really sad to see you guys go. It's been 4 months. Hope you learned a lot on this journey that we've taken through the realms of genetic engineering and science. I hope you learned a little bit. So I'm going to first give my plug, because that's what I'm here for, to sustain my salary and to sustain your learning. So so this is a course that teaches you genetic engineering for a span of a couple months, and it teaches you anywhere from a beginner stage of pipetting to calculations to weighing things out, all the way to transforming bacteria, transforming yeast, and genetically modifying and showing you glowing concepts of organisms. We are offering a Biohacker 102 class, which is up online. We're taking reservations for that. Class starts May 2nd. So if you're interested in taking the 101 or the 102, feel free to go on our website, www.the-odin.com, where we sell our classrooms. And what it comes with are options to choose between different levels of packaged experiments. So if you've already taken the 101 course and you want to take the 102 course, it's a discounted price because you have the essentials such as pipettes and tubes and stuff like that. And we'll send you just the perishables, the bacteria and stuff like that. So the 102 class is great. Josiah and I are developing the course right now. We are going to go through a lot in that course. We're going to go through pretty much the how to manipulate bacteria or how to manipulate genes. And then we go up to the point where how to design them, how to order them online for yourselves, and then how to transform them. And then we look at the results and sequence our gene of interest. And then we go through quite a bit in-depth form of bioinformatics. So if you really like the portion where you saw the protein, 3D modeling, you saw the sequencing of DNA, that is something that we're going to be doing a lot in the next course. And it's really helpful. I think it's really the bulk and the meat and the bones of genetic engineering, because what you learn in the 102 class or on the 101 class is how to manipulate your surroundings, how to manipulate bacteria, how to use simplistic tools such as pipetting. So the 102 class is really what you're trying to aim for sure. We will develop classes in the near future about plants and transhumanism, how to make yourself live longer, things like that. But we're kind of in the works to develop those classes in the next year, and it is your guys' support to support us. If you want more classes, the success of this class is based on your interest. And we always want to harbor that and keep that in mind. So, something that we always strive for. So That is up and coming, the 102 class. And then if you're interested in meeting us, we're going to all be in Vegas for Biohack the Planet 2019. It's a great time. This is our 4th year, but we are moving to a brand new location. We generally did it out of the Bay Area, but this year we're going to be moving to Las Vegas. We have a few confirmed people that will be there. It's kind of on the hush-hush, but yes, a few celebrities will be there. Definitely a lot of biohackers are going to be there. Josiah and the whole crew at the ODIN will definitely be there. So yeah, it's a science conference. I would check it out on YouTube or on our website, www.biohacktheplanet.com. And there's lots of talks of, between ethics, biohacking, different projects that people do. Lots of prominent biohackers have been featured, talking about their work. It's a good time, and we just get fucking drunk. So hey, I'm gonna take that wherever I go. So, all right, so those are my plugs. And yeah. Today, I'm going to kind of go over, just briefly go over like how your experiments are doing and what the CRISPR experiment kind of looks like. Unfortunately, Josiah is not here today or this week. He is on a business trip to the Dominican Republic. And he's actually doing quite a bit of filming there. So be sure to check that out in the future if you follow him on social media. He has a lot to say about medicine and how to practice gene therapies outside of the US. So it's a really interesting topic. He'll definitely talk about that. So without ado, I will answer questions about, or actually, let's kind of dive into the CRISPR portion. So everything has been leading up to the CRISPR experiment. It's what we want to do. It's what genetic engineering has come up to, and that's why we exist. The ODIN exists. So we sell our kits, our CRISPR kits, online on our store, and it just provides everything you need as far as this class goes. And so the main project is to genetically modify E. coli to express Cas9 and to be able to grow on specific selective media. And so it takes you through the steps of making competent DNA, transforming the DNA, inserting all the parts such as the plasmid, the template DNA, the Cas9, all inside of the mixture, and then transforming the bacteria and then plating it and then seeing results. So it's very simplistic. We've already done this because we've done it through the yeast GFP bacteria, or the yeast GFP kit, also the bacteria collar kit. So now this is just taking a step forward and inserting like a Cas9 mechanism into it. So that's when you can see genetic engineering take place. Okay. T. Williams, in 102, will we learn how to edit bacterial genomes to produce human proteins? Example, making human insulin using bacteria. In the 102 class, you will not be producing human proteins. You will learn how to edit bacterial genomes to do other things. I don't think in the syllabus it's very clear about what we will produce. So I think it's going to be like a collaboration of the class. So we will speak in class, okay, like, this is a bacteria of interest. What do we want to make? And it'll be more of a collaborative form to where all the students will pitch in like, hey, we should make like this protein, or hey, we should try and do this. So human insulin is definitely a very big one. It's definitely manufacturable by bacteria or yeast, which would be really interesting. So if you join the 102 class, you can definitely collaborate, and we'll go through all the steps of making this product a reality. So that's going to be really cool. And then Tara Williams says we'll be doing stuff with just plasmids via CRISPR for more permanent effects. Yes. Stay tuned. Okay. All right. So that's kind of like the CRISPR experiment. I know it's very vague, but it's all online on your online protocols through Google. And if you're interested in taking the course and being like, okay, this is like the big shebang CRISPR thing, and you want to check out more, you can definitely go to our website, www.the-odin.com. It's completely open sourced, our protocol, so you can go ahead and take a peek at that, see how the mechanism works, and see if this class is right for you, maybe. Yeah. Okay. So I kind of want to spend the next like 15 minutes-ish just to ask what you have learned. You know, I love chatting with students online. I've definitely friended a bunch of you on Facebook, whoever wants to take my friend requests. And I've been chatting with a bunch of you, and it seems like a lot of you have something specific in mind that you want to like genetically modify or work on as a biohacker, which is really interesting. I've heard anything from coral to Jeff's Pokemon to modifying like bees to pollinate better. So like I just want to know what you guys learned from this course, or what kind of things you took away from it, like reading papers or some of the guest lectures that we've had, or maybe some of the skills that you learned. I just want to see where it'll take you and how it'll affect kind of like the future of how you think about genetic engineering and how you're going to apply that. So any takers, anybody want to offer up something that they might have learned? Yes, Mr. Smith, you are kind of a big deal. Yeah, I totally agree with that. Come and see says, I'd like to modify the size of a dog, make a breed smaller, and make it slow shedding. Will I be able to learn these skills with future classes? Come and see. I don't know if you've ever heard of a biohacker named David Ishii. He's trying to genetically modify dogs to glow in the dark. You could definitely reach out to him on social media. He's definitely available on Facebook, and he'd love to talk to you about that. He has a different thing in mind for genetically modifying dogs. He wants to make them bigger and more wolf-like rather than make them adorable and non-shedding. If I were to genetically modify a dog, I would want to make them allergy-free, like hypoallergenic, because there's only a few dogs, maybe like 5 dogs that exist in nature that are hypoallergenic. But could you imagine if you could get rid of people's allergies to them? That would be kind of cool. Yeah. In the scope of the class, in the 102 class, we'll probably not learn how to make like these things. In the future, if these classes keep up, we'll definitely add like a animal bio course to our repertoire. But for now, we'll just see. Sub 3 learns more of this CRISPR thing. Sweet. Awesome. CRISPR says, I learned what the important skills are. Very descript. Jeff learns people are really against genetic engineering, even people who work in biotech with PhDs. I wouldn't say genetic engineering. I would say some people are against biohacking because they think it's unsafe. But if you've ever worked in a lab in industry, things are not generally safe, even though they're OSHA approved and BSL like 1 or 2 labs that get lots of like testing done, and and people show up to their place, it's not very safe. A previous biotech company that I worked with, not going to mention it here, yeah, we never wore like lab coats or goggles or anything, and we just kind of like did whatever we wanted. And then when we had investors come, we all put on like lab coats and then like goggles just to make sure that we look official. But you know, it's not safe, and that's why people are so afraid that like biohackers will do something unsafe. As long as we're practicing safety standards and talking about ethics and having a conversation to keep safe, safety's priority. So I don't think it's an issue, really. Tara Williams said, learn things that can be relatively easily transfected with simple heat shock treatment. Surprised me because in school we learned a bunch of chemicals that can do the same thing. Yeah, chemicals were like such an old school method on how to genetically modify things. But yeah, just a simple like temperature change can prove the lipids to be porous and then insert DNA. It's pretty simple. Matt Barnes said, I joined to hack together glowing beer or mead. I pretty much got all the info I need. I just need to work on deadlines out of the way. Yeah, so I think the glowing beer project is really crucial. We absolutely love it here at the ODIN. Peter works on genetically modifying yeast to glow in the dark. And we've been working on hazies to make sure that it suspends correctly within the beer after you brew it, because yeast, they die after you brew it and after you bottle it. So we've been kind of working on ways on like making hazy IPAs, making like sours, open fermenting, and trying to genetically modify bacteria and yeast that way. So it's a big process for sure. Yeah, Christopher Henningsen says pipetting and streaking, etc. Didn't realize how important these basic things are. Yeah, it's a really basic technique, but we don't really think about how to do molecular biology this way until we actually grow things and see things that are grown from it. So yeah, it's very important, and I'm very glad you learned those things. Pete says, make all the beers glow. Fuck yeah. High five. Christopher Henningsen asked, the heat shock thing probably helps to have an incubator for that, right? Not really. We have a water bath, and I've seen people like boil water on the stove and just get it to right temperature and consistent temperature, and they just stick their DNA in and pull it out. You don't necessarily need an incubator. All the stuff that we're doing is provided to you in the kit, and you can kind of like hack your way around a kitchen, because a kitchen is your lab, and it's pretty simple to manipulate a kitchen to be a lab. Matt Barnes, you started beekeeping? No way. I'll have to TM you, because I'm really interested in beekeeping and like making mead out of that. That's so interesting. Huh. It's pretty cool. Yeah, there's, like as I was chatting with a bunch of you, there's a lot of interesting ideas, and it's great to have a community like this, especially in the biohacker community. A lot of us are spread out in the world. We have so many biohackers that are interested in this hobby. And, it's kind of a really interesting network, because if we have an idea, we have a person to like that can manufacture DNA. We also have like a person in mind that does sequencing of DNA. So I think I've seen a lot of questions come up like, hey, should I buy a sequencer? Hey, can I sequence things in my house? Reality is sequencing is super expensive if you do it on your own. First of all, you have to buy the sequencer, which can cost thousands of dollars. and then your reagents and things like that, which you can't really make because it's very proprietary to the sequencer itself, and plus all the flow cells. So that can actually ring you up even more than the sequencer that you just bought. So there are places such as biospaces that do sequencing, and even like companies, in the Bay Area, like what Josiah was mentioning with those different sequencing companies, they do it for cheap, man. There's no reason why you should buy a sequencer. If you're curious on how to like sequence things, it would be a good idea to practice it. But like for the practicality of like, okay, I want to sequence like, yeast from beer, like I would highly recommend you go through, a service provider, which is like much cheaper. So there are points of contact in that. And then there's like there are lots of biohackers that know a lot of have a lot of knowledge about certain organisms. Like I just mentioned, like David Ishii is super into dogs and mammalian cells. He's probably a good point of contact. Sebastian from New York, he is a plant expert. There's lots of points of contact for that. If you just join the Biohacking and Genetic Engineering Design Facebook group, it's pretty accessible. Everybody will answer your questions as long as they're pretty like reasonable. So yeah, there are lots of resources, and I think I posted a big comprehensive kind of like PDF about where to find information, how to build a lab, how to get introduced to things, people to talk to that might be interesting. So even after the scope of this course, I highly still recommend you still go out and explore, even if you're not taking our 102 class. It's just a good idea to see what's out there, to develop scientific ideas in your head. And then hopefully, you can see light for, whatever you're trying to work on. I think that's what we're all here to do. We're all here to encourage you to do, to make something, right? Okay. So people have been asking me what's going to happen to the Facebook group. So this the Facebook group will still stay your Facebook group. We'll probably just change the name to the ODIN alumni. So you guys can still find each other, still talk to each other. Yeah, and share some really cool things happening in the biotech realm. You guys have been doing like a great job with that. And hopefully as we add alumni to the course or the Facebook group, it'll kind of grow and people will be able to mentor people there, have great ideas, collaborate, just things like that. So yeah, I'm pretty excited about that. Jeff Shu says, I already signed up for 102, but in case people didn't know what they expect to get out of 102, for example, after 101, can we expect physically, genetically engineering bacteria and yeast basic biostuffs? So the 101 is the basic biostuffs, and the 102 is how to manipulate those biostuffs. Yeah, we'll take you through the whole process of making a gene from scratch and inserting it into a host of our choosing, and then it'll make something at the end. And then we'll be able to quantify and do a little bit of bioinformatics, a little bit of sequencing. Yeah, lots of important things in 102 that you couldn't get in 101. Julie Miller, where can I find your PDF? If you are not on the Facebook group, I can certainly post it, email it to you. If you just, you can email me at esther@the-odin.com, ESTHER the-odin.com, and I will definitely send you the PDF, or if someone wants to hook her up with a PDF, that's cool. Yeah, Come and See says, I'm behind on my experiments with 101. Can you email me? Can I still email with questions after the class ends? Yeah, of course. I love hearing from all of you guys. Yeah, you can certainly email me. You can message me on Facebook. I will definitely give you some more information and provide technical support, because I know that people are still behind. I'm not like here to like, you know, leave you high and dry and just be like, bye. But no, I'd love to help you out. Even if it's not about the experiments, feel free to message me about like, hey, I'm trying to get my master's in this, or my PhD, like, or hey, like, I'm thinking about starting to go to a biolab, or like, I'm starting to do some like genetic engineering projects. Like, what do you suggest? I am definitely a great point of contact to just speak about anything. I'm pretty available readily. Yeah, so if you're one of those people that friend their instructors like after the course, that's totally fine. Okay, so yeah, our ending time is near. This is not really goodbye. It's more like an encouraging, like, you know, thanks for sticking around, hanging out, being our guinea pigs. This was our first ever class that we launched online. We really love watching you guys like grow and figure out how to do something so complex in a very short period of time. So I thank you. I thank you for, you know, just like dealing with our bullshit with shipping and technicalities like that, but it was really difficult to scale like 150 students from zero. So yeah, and you know, it's really up to you if you guys want to see this continue more with all these classes. There's nothing out there right now on the market that can market biotech in your home. And so we want to really do a good job on providing you with this service and, you know, hook you up with a community that understands and grows with you. So, yeah, it was such a pleasure just like meeting you. Just wanted to give a shout out to Jill and Pete. They've always been helping sit in the background, answer some of your questions, you know, troubleshoot anything that you've always asked about. And then, you know, Josiah and I are always readily available to talk to you guys. So, yeah, Pete says, you know, you got some cool tools now, but, you know, don't lose that knowledge, because it's a very unique piece of knowledge, and you can definitely utilize that to make some cool shit in the future. So, yeah, make sure you guys reach me on social media, and, you know, thanks so much. Yeah, 9897 SVXBG. You will have longer access to the Google Drive. So right now, you can access the Google Drive. You have access to us. In the future, you have access to these YouTube videos that will be posted online. Yeah, so, and then like the Facebook group will definitely live on. Yes, so we're not taking away anything from you guys. It's all the resources are there for you to keep and to refer back to and learn from. If something happens, just make sure you email me, and I can definitely get you the documents so you can save it on your hard drive or whatever. So, yeah, thanks a lot, Matt. Thanks a lot, Mr. Smith and Chris. Yeah, it was so great. Thanks, 97. It was so great chatting with you guys. Hope to see you guys soon. And yeah, hope to see you at some conference or something. That would be really cool to meet in real life. All right, take care, you guys. Bye. Burp, burp.
All right, that's 13. Last class for 2 weeks.